Hello, everyone. Welcome to Food Safety Virtual Office Hours. So today's office hour is focusing on how to select the correct lab for testing. Now, I'm going to preface this with I was a bit hesitant to throw out this topic because at least in some cases, a tendency to blame the lab if a result turns out positive for Listeria or Salmonella or not pass whatever test it is. And that's the last thing we want to imply here today. However, and that's important, that sometimes we see challenges with labs that offer or do tests relevant to food safety. And I think it's important for everyone to sort of recognize those features and try to minimize the risk that your lab isn't doing exactly what you needed to do. And that's what this session today is about. Today we're gonna have a presentation that will be presented by three people: Anna Schumann, a postdoc at Cornell; Catharine Carlin, Vice President of Technical Services at IEH; and then Dan Stoeckel is a Senior Extension Associate with the Produce Safety Alliance. Will talk to us more specifically about labs for water testing. And after we're done with that, we're going to entertain some questions, which I may or may not weigh in, depending on how I feel at that time. We got some pre-recorded questions or pre-submitted questions, but if you have any other questions, please add them in Zoom and we'll take a look at them and prioritize them. With that, I'll turn it over to Anna to start us.
Thank you, Martin. As Martin said, today we'll be talking about how to select a testing laboratory, and that are some considerations for food matrices, environmental swabs, and agricultural water testing. And so, in general, I don't think I need to kind of say this for anyone on this call, but testing matters because we all care about food pathogens. There's 48 million cases per year of estimated foodborne illnesses in the US. That comes with an annual burden of about $75 billion, and has a leading impact of about 1 in 6 Americans get sick from foodborne illness every year. And so why do accurate results matter? There is a high cost associated with inaccurate results, and that goes for both false negatives. So you might get a test that's negative, but the result would be positive. That comes with an outbreak risk, a public health risk. It comes with product recalls and then obviously brand damage. However, false positives also come at a cost, meaning you get a positive result that is actually negative. You might have unnecessary product destructions or recalls. There might be costly corrective actions, unwarranted negative perceptions, or supply chains disruptions. So a lot of different things to consider. And so first we'll start with the food matrix and environmental swab testing, and then later Dawn will go into the water testing, as Martin already mentioned. So first of all, why is bacterial testing so challenging? Bacteria are often present at low levels, so there might be some growth challenges associated with the matrix inhibition or sublethal injuries. There's a lot of background flora, so it might be hard to identify the specific bacterial pathogens you're testing for. Think about Listeria, Salmonella, etc. There's a difference between detection and isolation. So modern rapid methods offer faster detection. So basically giving you a result, is this specific pathogen present or not, versus isolation, where you actually get a colony that and your specific pathogen that you can potentially further analyze. So thinking about cultural confirmation here, and this can be lengthy and potentially subjective because it involves a lot of looking at plates and identifying very specific colony morphologies. And then we also have to consider the diversity of bacteria. Listeria species, for example, are highly diverse and have different phenotypes that need to be accounted for when you have a test that is supposed to test for all of the different species, similar to the Salmonella serotype, where some may have atypical characteristics, so making it very challenging. I'll turn it over to Catherine.
Hi. So thank you, Anna. Just reviewing, the two general approaches with respect to how a sample is analyzed. So this is specifically talking about food microbiology and pathogen detection. So there's the culture-based or reference methods. This will be your FDA BAM, your USDA MLG, and then there's your rapid detection. So there's literally hundreds of options out there today. It's important to know how to evaluate and differentiate those options. But with respect to pathogen detection, it's either a rapid method or culture-based. And the trend is very much towards rapid detection because it offers that screen of the enrichment. So then the lab can focus on only those enrichments that are presumptive positive, so they can screen out or filter the negatives. However, there are still labs that are primarily running on culture-based reference methods. And it's important to understand while you don't need a lot of expensive equipment to run these methods, you do need a lot of experience. So it takes years to be able to look at a plate and differentiate visually what is highly likely to confirm positive versus background flora, which is potentially an atypical colony, versus the rapid methods where the workflows are quite simple, the turnaround time is fast, and the level of expertise to run these methods is getting easier and easier. Some of them have just one or two steps, and then you run it on an instrument that does the result interpretation for you. Importantly, if a lab is offering the culture-based reference, there are many steps to these methods, and any modification to try to simplify or reduce the level of complexity is considered a major modification and should or needs to undergo validation. So just understanding if you're running a reference method that it should take several days, if not a week or longer, for that to finalize a result, whether it's positive or negative. And a note on presumptive positives. So Anna talked about false positives and false negatives. I think it's a common theme in the industry to bin together a false positive with a presumptive positive that doesn't confirm. And these are two distinct results. So with rapid detection, so there's a screen, often a molecular screen, you're looking at DNA. It's a highly sensitive test. And subsequent to that, it can be very challenging to isolate or tease out an isolate that you can then call a confirmed positive. So there are instances where you have a presumptive positive that does not confirm, but that does not necessarily mean it was a false positive. So it's important to have that distinction when evaluating your results. It is also important to understand how that presumptive positive was generated. So if it's a rapid method, it's a highly sensitive method. It's very likely that the target was there. If it was a culture-based method, there is some level of subjectivity, whether or not that is truly a presumptive positive at that point. So if you're taking action on a presumptive positive, if you're starting a cleaning regimen, for example, it is really essential you understand how reliable that presumptive positive is. Again, if it's a culture-based, there is a higher probability that it is not a true positive versus a rapid detection.
Okay, so sorry. And we're now going to go into some of the key aspects when evaluating a testing laboratory. And Catherine and I will be going back and forth a little on this one. And so the four kind of key things to consider is laboratory accreditation. So is your laboratory accredited or the one that you're considering of using? Are the testing methodologies validated? Also assessing the internal procedures. So basically, are they still doing the things they say they're doing and that they're accredited for? And appropriate use of process controls. And so first of all, accreditation of the testing laboratory. The global standard for lab competence is the ISO/IEC 17025 standard, and this accreditation happens via accreditation bodies. There's different ones. For like, I'll give an example later on what kind of an accreditation certificate looks like from one of these organizations. But the nice thing is all of these different accreditation bodies have websites where you can look up accreditation certificates, and you can even use those as kind of a search tool of what state you're in, what type of test you're looking for, and then it gives you a list of accredited laboratories. Then specifically for food, there's also FDA LAAF, which stands for Laboratory Accreditation for Analyses for Food. These are FDA recognized accreditation for food testing laboratories. And then USDA has something similar, which is called ALP for Accredited Laboratory Program. And that's just based on what food is regulated by each of these. So for USDA, it would be meat, poultry, and egg testing. And that isn't just for microbiology, but also food chemistry and chemical residues. I'll give it over back to Catherine.
Thank you. So again, looking at the scope of accreditation, what is a laboratory? Not only what standard are they accredited to, but what is on their scope of, I guess, quote unquote, approved method. So again, there's that gold standard, and these are going to be your reference methods. And one of the, I guess, values is these are already validated. They typically have a claim broad range of foods or all foods, meaning that they can be run without additional evaluation of what matrix you are going to be submitting. So if you're running the rapid method, it's important to understand what that method claims. So not every method is accredited for every matrix. And oftentimes we're seeing that rapid methods need additional validation or verification data to support the specific matrix that you're submitting. And minimally, that just evaluates how the method performs with your matrix. So Anna mentioned inhibitory properties. So that's a key point to consider in these validations. Is there going to be inhibition that's going to lead to a potential false negative result? So in those cases, methods may need to be modified in the sense that you need to do higher dilution ratios. You might not be able to test the analytical weight that you're requesting, or it needs to be done in multiple subsets. Now, AOAC is the main standards body that's used in the US. It is internationally accepted. But what many people, I think, fail to realize, just like with ISO 17025, which we'll get into, this is just the primary or the main distinguishing factor. But there are different levels. So within AOAC, there's a PTM and OMA. So these PTM is the initial single lab validation, which means that the statistics that were performed were based on a set of 20 samples. So just understanding the potential limitations with that, and then an OMA level validation has gone through multiple laboratories to assess how different labs would perform, how this method performs at various labs under different conditions. So looking at the scope, understanding what the reference method is, what is referenced on that scope, so you could have a reference method or a rapid method, and then having access to that method and how it is run is important. So for AOAC PTM, that's probably one of the most popular. It is a publicly available website. So you see an image here on the right. So you can enter, I always start with the analyte of interest. And then if you already know what method you're running or you're interested in, it'll have a certification number, and you can input that, and it will lead you to the validation report. And you can look at the matrices. And I think it's important that you do that because you can see that while the method has passed and is certified, there are some level of disagreement that is permitted. So you can have, for example, up to 3 false negatives and the method will still be approved. So knowing that there is a potential, for that method to generate some false negatives or false positives is important to consider when you're selecting your method. Next.
And so now that we've kind of talked about both of accreditation of the lab and scope of accreditation, I made a very generic accreditation certificate here. This is A2LA, which is one of the accreditation bodies. And the way it would look like, you have the name, you have like accredited laboratory here, the company name, city, state, and then what it's for. It can be biological or chemical. You will get a certificate number when it's valid to. So also important to pay attention to when was this, presented, and then how long is it valid to, and then it will also always come with a scope of accreditation, and then here you can see the 17025. Again, it will have the company name, address, and then a contact for the lab, certificate number, and then to make it a little bit more clear, you will have the test that this lab would be running, for example, coliform counts, listeria detection, salmonella detection, standard plate count, and then it will here in the second column say what the test method is. So here you can see it's FDA BAM, so like one of our gold standard cultural methodologies. And then there's some references here. And then most importantly, if there's any changes that were made, they should be listed here, at least in a footnote. And ideally, obviously, these should be validated changes. And then sometimes, besides the test, it could potentially also list what analyte it is, which for FDA BAM, for example, it is very broad, so it's not specifically listed here. But like Catherine said, for some of the PTM validated methods, for example, it would specify what analyte or what matrix it is.
So it's common when a lab is running a method, they should have their own written SOPs for that method, just outlining how they run that method in their hands. So for example, if you look at the FDA BAM, it'll say for salmonella that you need to incubate your tubes in a circulating water bath. And so I would look to the local procedures that it says that they use a circulating water bath, and then there's other criteria that are specific to how the lab is running it. For example, do they have separate water baths for samples that have a high level of positive versus low level? Do they clean the water bath? Do they add sanitizer to the water bath? So these are all details that you would expect to see in a local procedure, in addition to, are they following all steps of the method that they reference? And just to go back to Anna's the previous slide where she showed the method that is referenced, you will also commonly see a link or a reference to the local procedure that is used for that method, if that is what is referenced on your COA. So there should be that information on the scope, and it should also be a method that has a level of detail that someone new to the method can access it and understand the various steps that need to be done, and they're not asking someone. So it's not a tribal knowledge piece of paper that doesn't have a lot of information. And there also should be training documentation associated with that method. Usually you'll find a checklist of the critical points of the method where someone has signed off that is a designated trainer. So it's also important to understand at the laboratories, who is that person that is assigned training, because they should be a higher level employee not someone that just happens to be next to you that day, telling you just take 10 microliters and do this. It's someone that is certified to do that training. Next. And another point is the process controls. So a lot of times the volume of samples going through a lab, the majority of those are going to be negative. And depending on what commodity you're working with, it's rare to have a positive. So what ongoing qualification is there that the lab is maintaining competence, not just in the method, but in their ability to not cross-contaminate? So positive or process controls are important, not only for that point, but also how does a lab maintain that, or how do they demonstrate ongoing aseptic technique? So in my experience, aseptic technique or poor aseptic technique is a leading cause of laboratory contamination. So you want to see that they're continuously and regularly running negatives next to positives. And they're also including atypical morphologies in that assessment. And for the positive control, it is very commonplace now to use a genetically modified strain. So it's most often a insert that allows for fluorescence on a plate. It doesn't occur naturally. So you can easily distinguish between a positive generated from a client sample, excuse me, or a food sample and one generated from the lab. So the lab should be able to quickly and easily distinguish whether it is their internal control strain, which historically has been a leading cause of lab contaminations, thus giving that lack of confidence potentially in suppliers that it's a laboratory error. So the laboratory that you're using should be able to give you high confidence that they have generated a presumptive positive and that it's not a laboratory issue. And it's key not only that they can distinguish the positive from a wild type, but that they can show regular and repeated measures of good aseptic technique. So I've seen going into labs that they will have, positive controls that are so removed from the process that it's actually a separate process. So it's important to see that it's integrated and regularly being performed and rotated through all employees. Next.
Yeah, so kind of getting to the comparing labs, what to ask, and this is kind of getting into kind of the conclusion piece of it. So one, is the method on their accreditation scope applicable to your food matrix? And that is, like Catherine was talking about, more for those PTM methods, the culture-based ones, the FDA BAM, they usually cover a lot of different matrices. Then two, are the SOPs detailed, validated, and available to you? And these should minimally be available on site. And that again goes back to how easy is it to follow the protocol? Can someone new follow it? Or are we relying on word of mouth? And then how do they handle and report presumptive positives? When do you consider presumptive positive to be questionable? And then are there mechanisms where the lab can go and further process a presumptive positive, for example, for those cultural-based presumptive positives? And then lastly, what measures does the lab take to control laboratory contamination or cross-contamination? And so overall, I think it's very important to have confidence and trust in the lab. It's kind of like a process that goes into both ways. So you want transparency. Labs should openly share methods, scopes, and control data. Welcome visits and audits. Expertise should be maintained in-house by you or contracted for testing laboratories and methods, so that you can determine, do we trust a lab? Which lab do we want to pick? And also important to have high standards and also setting clear expectations, because accurate results protect public health and also your brand.
So, wrapping it up with some key considerations. So ISO 17025 is globally recognized. It's the predominant laboratory accreditation certificate that labs will attain. But just know that it's just the beginning. So it is broad and detailed, but the laboratory is only audited one every 1 to 2 years. It's a brief audit where they come in, and sometimes they're not technical experts. It's checking boxes and looking if they have this. Do they have the layout in place? Do they have a training program in place? So yes, it's good, but the key oversight should come from you when selecting that lab. I think it's valuable to have a set of criteria that you want, a checklist for yourself to go over. Um, it's very informative to do a traceability exercise on your sample. So the point when it gets to the door till when it has been reported to you. So understanding how it was managed at sample receiving, you know, traceability to the media that was used, to the technicians and their training, and to the equipment. Is the incubator mapped? Are the temperatures um, measured at the appropriate frequency? So visiting the lab, ideally just like you would any supplier, is important. And having somebody on your team that understands the method that either you're being told is being run or that you've requested, and is that method being run appropriately. So I think a lot of people default to, I'm running an AOAC method at a 17025 lab, and so everything must be in place and all checks and balances are there. But as we've seen in the past, that's not always the case, and it's important to have that level of oversight of the lab, especially if you're going through any type of event, like for example, if you're seeing an increased frequency in your positives, to know what measures that they have in place and to have high confidence in those results. So minimally having someone that has some microbiology expertise is important. I've had several conversations with people, and the first thing that they say is, I'm not a microbiologist. I don't understand these results. So having that resource is important. And in doing the traceability exercise, you learn a lot about how that method is performed. Whether there are any deviations or any potential indicators that something could happen in the future is important. So sorry, I just reiterate that when selecting a method, again, it's kind of overwhelming these days. Like, it's not an exaggeration to say that there's hundreds of AOAC approved methods out there, and they're not all the same. So understanding the pros and cons of the different technologies. So they're going to come at a different price point, and understanding what you're getting for that price, and understanding, how long have they been on the market. If it's something that's been established for many years, there's, value in just asking others, what their real world experience is, because these methods parameters are established in a laboratory setting with artificially contaminated foods, which is close to what you might see in the real world, but it's never going to be exactly the same. And especially for environmental monitoring, because it's very difficult to mimic an environmental condition in a facility versus in the laboratory. To validate an environmental testing program, it's a 1 foot by 1 foot square sterile coupon that's inoculated with the target of interest and then swabs 24 hours later. So very different from what you would see in a plant environment. Um, okay, thank you.
I will take over then. Thank you. And all that great information we just got is one reason why we have such popularity in this office hour series. Um, what we just heard was the matrix of things that a lot of operations in the food system need. I'm going to take us back to the base of the food system and talk about produce farms and agricultural water testing, because this series is so popular. And if we do have farms represented or people who need to know what farm requirements are, when we wanted to make sure that that was clear to everyone. Next slide, please. So farms are covered by the Produce Safety Rule, which is under the Food Safety Modernization Act. And the only testing requirement that produce farms have is for agricultural water quality. And I want to point out what's the definition of agricultural water, because it's not intuitive. It really throws people for a loop sometimes to realize that agricultural water does not mean water that's used in agriculture. Agricultural water actually means, in context of Produce Safety Rule, water that's intended to or likely to contact covered produce or food contact surfaces. So, for instance, drip irrigation under plastic for a tomato field might very well not be agricultural water, but overhead irrigation on a tomato field very much would be agricultural water. And one of the reasons is that if the water contacts the crops, it has a higher chance of any contaminants that might be in the water getting on that crop. Next slide, please. The next slide is going to talk about pre-harvest agricultural water. And pre-harvest agricultural water is mostly irrigation water. And in 2015, when the Produce Safety Rule was published, there were testing requirements for pre-harvest agricultural water. Those requirements were revised in 2024. The new requirements for managing agricultural water that's used pre-harvest for growing produce and now relies on something called an agricultural water assessment instead of testing. An agricultural water assessment uses a lot of the same processes and procedures as a sanitary survey, and it's still valuable to have test results as supporting information for that agricultural water assessment, but it's not required under the Produce Safety Rule to do any testing for pre-harvest agricultural water. When a farm chooses to do testing, they're as often as not going to compare the results they get to recreational water quality standards like 126 CFU or 410 CFU per 100 mils of generic E. coli. Next slide, please. It's a little more regimented in post-harvest water. And in other words, one common post-harvest use of water is washing your produce or fluming produce in the operation prior to packing. The standard there is no detectable generic E. coli in 100 milliliters. There's no flexibility on that. It's got to be generic E. coli. It's got to be no detect in 100 mils. And you may recognize that as being the same as the MCL for drinking water under EPA's Safe Drinking Water Act. A produce farm that is using post-harvest water and they're using untreated groundwater will do the testing at the farm 4 times 1 year, 1 or more times the following year if it's untreated groundwater. If they're buying their water from a municipality or other public water supply, those tests are done by the utility, but it's going to be the same tests, same expectation, no detectable generic E. coli. If it's treated water, then if that treatment is done with a validated process that will achieve the expectation or standard of no detect E. coli, then there's no testing necessary. Regardless of whether it is tested or not, untreated surface water cannot be used as post-harvest water in a Produce Safety Rule regulated, Produce Safety Rule covered farm. Next slide, please. So getting to what does the laboratory do for these farms? The sampling is often done by the farm, but the farmer doesn't always know how to do sampling using aseptic technique. The lab often provides a sterile bottle, and we encourage farms to find a lab that's going to support them, because the laboratory is not the farm operator generally is not a person who knows how to use aseptic technique. They're often not a person who knows what the expectations are for delivering on ice within 8 hours or whatever the hold time expectation is. We talked a lot about ISO 17025 and other accreditations. FSMA Produce Safety Rule does not require that the lab be accredited, but of course, it's a great idea to know that the lab's accredited, because you have some quality, you have some assurances of quality of the analysis. And accreditation can be required for some of the audit schemes. And then the third thing that the laboratory, when we're selecting a laboratory, we want to make sure that the lab is using FDA accepted methods. There's a list of both quantitative and qualitative methods because the only standard written into the produce safety rule is no detectable generic E. coli. Presence absence tests are acceptable in the FDA's list of accepted methods, but also quantitative tests. And especially when we talk about pre-harvest water, where we're comparing against recreational water quality standards, often the use of a quantitative method is appropriate for a pre-harvest water test that's used to support agricultural water assessment. On the right, there's a list of the methods that are written into an FDA fact sheet on what methods are accepted for produce safety rule regulatory monitoring. Next slide, please. So how does a farm find these labs? It's actually a fairly simple process. NECAFS, the Northeast Center to Advance Food Safety, has a food safety resources clearinghouse. And on that clearinghouse is a map. And many labs have voluntarily provided their information, including what's the method number they use, so that you can compare that method number against the FDA's list of accepted methods, and what days they accept samples, and along with their contact information. So it's a fairly simple process for a farm to find a laboratory that can meet their needs. And that's all I have for farm-based testing lab and what that farm would look for in that testing lab. Thank you.
Okay, thanks, Don and Anna and Catherine. So we're gonna go to the question part. I'm gonna